Today we have Professor Shilling Shan from Duke University for the Terasaki seminar series. Professor Shan is the Hawkins Family Associate Professor in the Department of Biomedical Engineering and the Director of the Wu Center for Big Data and Precision Health at Duke University. He is also the CEO of Silis Inc., a precision medicine company that recently raised 70 million Series A. He received his bachelor's, master's, and PhD degrees from Stanford University and the NSF Career Award at Cornell University. He is the steering committee chair of the NCI Patient Right Model of Cancer Consortium. His lab studies precision medicine from a systems biology perspective. He is also interested in cancer, stem cells, gut-brain axis, and infectious diseases. Floor is yours, sir.
Great. Mehmet, thank you very much for the introduction. And can everyone hear me?
Yes.
Oh, okay, great. Thank you. So I'll just share my screen. Great. Again, this is really a pleasure. Again, I've known Ali since we started the NCI Tissue Engineering Consortium. Actually, I hosted a meeting, I think, last week. And we actually were supposed to have a collaboration actually before when Ali was still in UCLA, but then COVID hits. So it's great to reconnect and also really kind of exploring this very exciting opportunity working with many of your folks. So today my talk is gonna be about kind of the various tools my lab has been developing to answer questions about tissue function in order to improve precision medicine. So here I just disclose I have two companies, IQBeing and Xylus. So what my lab is interested in is really addressing this question about how to make treatments more precise. As we often know, a lot of treatments, especially for cancer, are shots in the dark. So today I'm going to talk about kind of 4 areas my lab have been working on to fill in the gap and also to provide a holistic view of tissue function for precision medicine. I'm going to talk about how we develop functional assays to improve clinic cancer treatment. I'm going to talk about various omics tools, especially epigenetics we use to further understanding reprogramming the tissue. I'm going to talk about how to identify microbiota in the tissue and their function on the tissue and their impact on tissue function. And last, kind of a lot of our intravital imaging work to understand tissue and also development better. So first, I'm going to talk about the work we've done on the organoid work we did on improving patient cancer care. So as many of you know, cancers are a leading cause of cancer death, but big frustration is also the ineffectiveness of the current treatments. A typical scenario is when a metastatic cancer patients were diagnosed. They undergo this called a standard of care, where they receive first-line treatment, second-line treatment, third-line treatment, and their response rate to the treatment decreases progressively. And very soon they will run out of options. Either they are too weak to receive any more treatment and go to hospice to wait for their time, or they can sign up for clinical trials of new treatment. But now historically, the chances that their tumor are going to respond to a new treatment in a clinical trial is about 5 to 8%. So, right, if you go back to like a decade ago, genomics was hailed as the solution. And that gave rise to very successful companies like a Foundation Medicine and Guardant Health. It's all, $10 billion plus companies. And the idea is very simple. If we can sequence the tumor, we know what the right treatment to take. However, there's an elephant in the room, which is this 7% number. Right now, only 7% of the cancer patients actually benefit from genomic sequencing. And the reason is very simple. The majority of mutations are not druggable, right? The mutations that are druggable are kind of basically a handful you can count with your fingers. So, but the majority of patients will receive a sequencing, but end up the condition will go on with the standard of care. So how can we improve the cancer care for the remaining 93% of the patients? So here, actually, the reason I was a faculty at Cornell and I moved down to Duke, is reason to work with the clinician David Shi, who is a medical oncologist here, to really think about how to work together, an engineer with a clinician. Now, here, so this is a typical case, right? Where with those 93%, this is actually a single mom of 2 and had and diagnosed with metastatic colorectal cancer. David treated her with a frontline drug, oxaliplatin. 3 months later, the tumor progressed, switched to irinotecan, the second-line treatment. And meanwhile, she developed the first neuropathy, and then lost all her hair. She lost her job, and eventually, the tumor kept growing. And eventually, she was taken off the treatment and passed away several months later. And these are really the kind of patient cases that inspire us to do something better. Can we be better? So the technology Dr. Hans Clevers invented called organoids caught a lot of attention and generated a lot of excitement. Right about 12, 13 years ago in 2009, when Hans first published that you can grow tissues in these 3D matrix gels with the right growth factors, and these tissue cells can grow. Basically, the stem cells are able to self-renew and also to give rise to tissue-like structures. Now, over the past 3 years, there are kind of a slew of very high impact, exciting papers showing that the organoids can actually correlate with patient outcome very well. The numbers they often cite is about 90%, the positive predictive value is about 80%, means 80 to 90%, means that if an organoid responds to a drug treatment, the patient, in 80% of chance, the patient also responds to the treatment. And also this negative predictive value, right? And sometimes even 100%, which is if an organoid does not respond to the treatment, the corresponding patient also does not respond to the treatment. So we actually got asked by clinicians a lot when we in organoid meetings, like, hey, this is great. Why they are not being used in the clinic? And these are things that's preventing the organoid technology to be applied in the clinic rather than just a research tool. So what is a missing piece? So this is actually the latest number from Dr. Hans Clevers in his head and neck clinical trial in Europe. So for every patient, it costs $4,500 to derive these organoids and do the drug treatment. A major reason is it takes a long time because for clinical treatment, you are taking a small biopsy and try to expand those cells to a large number so you can do the different wells, different treatment, different repeats, right, different dosages. So take about 3 months, 2 to 3 months to establish them for drug testing. Less than 50% success rate. Now just imagine you're telling a patient to wait 2 to 3 months to receive treatment, and more than 50% of the time, you go back to the patient and say, oops, your organoid did not expand enough for me to do the assay, right? So, and last thing is, all those are also mainly done by hand. From operator to operator, there's a great variability. So it's not very reproducible. And obviously these cannot pass clear certification to be a commercial diagnostic assay. So each one of these limitations is sufficient to kill organoid as an application for clinical diagnostics. So what can we do? So here we kind of, my lab kind of, again, in collaborating with David and Hans, invented this technology called the Microorganospheres, where we can take a small piece of biopsy, encapsulate tumor cells with stroma immune cells into these tiny droplets of gels. Can be synthetic gel, can be matrix gel, where the tumors grow into organoids within its environment. And very rapidly, by day 6, we can dispense these little tumor balls or microorganospheres or moss into high-density wells, and using imaging-based assay to analyze their response, individual organoids' response to treatment, picking the best drug, and use that back on a patient on average in 10 days. So this is really, and because in the clinical flow, usually the metastatic cancer patients are put on treatment between 14 and 21 days. Oh, sorry. And what's made it possible, again, is this technology. We did a microfluidic technology. Again, there's several challenges to make this as a clinical grade. First is when you deal with biopsies, you need to have a very, very high recovery rate. What that means is because you get such a small piece of tissue, almost all the cells count, right? You cannot afford to lose 50, 70% of cells. So we have higher than 90% recovery rate. It means 100 tissue cells come in, 90 cells come out. These involving with eliminate the dead volume that involve inventing new membrane technology for better, more efficient and less toxic demulsification to recover these droplets from oil. There's multiple engineering marvels there. But anyway, the long story short is we were able to capture these patient tumor cells. And you can see that's kind of really caught everyone, right? Including Dr. Hans Cleavers, really surprised. These tumor cells actually form organoids very rapidly in these mouse environment. And from the hindsight, we think there's a couple of reasons. Number one is because of small dimension, it has a much bigger surface to volume ratio, promoting exchange of growth factors and nutrients into the organoids. And also second is we are creating a higher local density because patient cells need an environment. They need to talk with each other, cancer cells, and also with the stroma cells. Furthermore, but these organoids can be passaged generation after generation. And also using microwells, high density microwells, we can do high content imaging, tracking every individual mouse longitudinally and look at their response to the treatment. So using this technology, we're going back to the patient cases. So I remember I told you before about these colorectal cancer, metastatic colorectal cancer patients, and the response, when we talk about response rate, let's say 30%, it doesn't mean, right, the 30% of patients were cured. All that means is the drug does anything to that patient, right? Even just halting the growth. So you can see how ineffective right now the drugs are just being tested up, put on the patients. So what really inspired David and me, and again, we started company Xylus, is that patient, Julie, who I told you about. The fact of the matter is when we test our assay on her, when we used our technology, in 8 days, we knew she would not have responded to the 2 frontline treatment. But of course, at that time, there's no validation of our technology, so she went on to have those 6 months of treatment, validating that actually our technology was predictive. But on the other hand, it was heart-wrenching to see a patient undergo those field-type treatment while we have a technology in hand, knowing that she's unlikely to respond. So since then, we actually started a clinical trial. It was slated to start February last year, but got stopped by COVID-19 and resumed last November. And this was our very first patient. She was presented with the lung mets where we did a mouse. On day 8, we did a treatment and found her tumor is sensitive to oxaliplatin. On day 12, she was on the oxaliplatin treatment. Again, just to clarify, we did not alter her treatment, right? This is still a correlation study. We are just correlate with the response. But 6 months later, she's still on treatment and the tumor shrinking in response to oxaliplatin. And since then, we have been doing this clinical trial. That idea is guided by biopsy, generating mouse and do the drug treatment. And so far, we have done 23 patients. So first is 22 out of the 23, we were able to establish mouse and did a drug treatment. The only case that failed was interesting. She was presented as a lung cancer patient. And eventually we found out is that she actually had a rare type of neuroendocrine tumor. So it was not a lung cancer. And also on average, it took us 10 days from biopsy to finish the drug testing. So these are the first 8 colorectal cancer patients. Again, later on, we are actually expanding to all cancer types. But the very first 8 colorectal cancer patients, for the 4 that our mouse responded to the treatment, all 4 of them are still on treatment past 20 weeks. While for the 4 cases where the mouse were sensitive to the treatment, were resistant to the treatment, oxaliplatin, 3 of them were taken off treatment within 10 weeks, right? Finding that the drug was not did not have any effect on the tumor growth. The last patient is on week 12, and actually he is likely going to be taken off pretty soon, which I will show you in the next slide. And also, if you find other cancer type, the message is very consistent that our mouse predicts patient outcome. So that was the lung patient I will just mention to you. As you see that, although he's still on treatment on week 12, the latest CT scan showing that his tumor is still growing. So expect him to be on treatment off treatment pretty soon. And based on this data, now we are starting a newer guided trial. In this case, we are actually targeting metastatic cancer patients that pass third line treatment, right? These patients right now have very limited choice. And we are again doing our assay, but this time we are guiding their therapy. So that they will receive the therapy based on our assay prediction. And this is led by, again, my collaborator, David, but also Dr. Scott Kopetz, who is a leading GI oncologist. He's the deputy chair at MD Anderson, leader of the Moonshot program, and also is the chair of the NCI colon cancer task force. And the primary objective is to improve these patients' overall survival or time on treatment. Right now, for these patients, they have a miserable outcome. The average overall survival is 1.8 months. That's it. So we actually had assembled a clinical board with clinicians both from top institutions like MD Anderson, Mayo, UCSF, but also with community clinicians from community clinics. Right? We asked the question, how much improvement our assay need to provide the clinical meaningful results for everyone to say, look, I have to do this assay to guide my treatment, because otherwise it becomes unethical to give a patient random treatment just following your guts. So the goal is to improve their overall survival past 5 months, and that will be a very convincing result. So furthermore, besides using the mouse to guide chemo and targeted therapy in the clinic, we also asked the question, what's in those mouse besides the tumor cells? So interestingly, when we did a single cell sequencing, that was actually recommended by Dr. Aviv Ragev. Some of you know, right? She was kind of the mother of a single cell, director of the Broad Institute, but also now is the director of research at Genentech. So she said, why do you guys do single cell? So we did a single cell sequencing, and we found out that, interestingly, our mouse really preserved the immune environment very well within the first 14 days. You can see the different components are still there, and you cannot really tell them apart. Now, furthermore, our mouse actually recapitulates the immune suppressive environment in the tumor. So here on the left, again, this is another case of another lung cancer patient. So the purple is tumor cells, green is lymphoid, basically T cells and B cells, blue is myeloid cells, including natural killer cells, macrophages, and dendritic cells. And this is a fibroblast cell. So you can see, if you look at the checkpoint inhibitor, as many of you know, right? So these are the targets of immune therapy because they create a tumor suppressive environment. For the PD-L1, they are mainly expressed in the tumor cells and the myeloid cells. For PD-1, it's expressed in the lymphoid T cells. TGF-beta is also have this T cell, but also some tumor cell expression. So you can see that in the single cell analysis revealed that our mouse recapitulate this distribution pattern very well. But furthermore, this provides the very first assay to actually test this immune therapy directly on patient tumor. So right now, pharma companies, when they have immune modulatory drug, what can they do, right? They can use it to treat mouse tumors, or they had to put into patients, because there's no models right now out there that can both capture the patient tumor and the patient and the immune environment in that tumor. So our mouse now provide a very first such model. And in this case, this is from a lung cancer patient. You can see the lung tumor organoids growing in this tumor environment. The other cells in the droplets are stroma cells and immune cells. Now, green is the cell death signal, but you can see there's minimum because these tumor cells are growing. Now, as soon as we add this checkpoint inhibitor, you can see the immune cells the original immune cells in the tumor environment started mobilize and attack the tumor organoids from the peripheral until penetrating the entire tumor and basically killing the entire organoids, showing how effective, and that patient actually did respond to the checkpoint inhibitor therapy, which is about 20% of the lung cancer patient. So with that, really, we are working with Dr. Scott Antonia, who's the director of the Duke IO Immune Oncology Center, but also he's the one who conducted our original clinical trial that made immune checkpoint inhibitor the standard of care and NCCN guideline for lung cancer, both small cell lung cancer and non-small cell lung cancer. So here, in this case, we actually now testing combo therapy, nivo plus ipi, to test whether our mouse can predict patient outcome. So these I show you that this mouse technology used to improve patient diagnostic, right? To try to predict patient outcome, so help bring the right treatment to the right patient. But then also we are finding that technology also meet a very critical unmet need for the therapeutic development, especially for pharmaceutical companies. Now, a lot of you know drugs are expensive, right? But kind of put into the context, here is some numbers where 95% of the oncology drug fail in FDA sanctioned clinical trials, right? Only 5% made through the clinical trial. For each failure, so majority of them fail in phase 1 or phase 2. On average, it takes 4 years and $600 million for each drug to fail, right? And if you put that number onto all the drugs, each successful drug costs $1.7 billion. And a large part because of this high failure rate. So you can imagine if we have a better model that allows farmers to predict the patient cohort who will respond to the treatment, we can significantly improve this pipeline and de-risk this costly process. So here we actually work with a major pharma to test their very new biologics, so antibody drug. So this is called a TCB, or bispecific T-cell antibody, which is kind of all the rage right now, where one end binds to T-cells, CD3, and the other end binds to the presented tumor test the antigen on the tumor cells. Now, interestingly, in this case, right, again, as I said, all they can test it, and they can't even test in mouse because the drug recognizes human antibody, right, human antigens. So there's a tremendous risk to take a drug like that to clinical trial. In fact, their competitor is a smaller biotech. It did take a similar drug to clinical trial. I happen to know because Duke was one of the trial sites. And after 2 years and tens of $millions, they get very mixed results. So here with the farmer, within 3 months, we were able to test on more than 30 lung cancer patients. And again, identify patients who respond to treatment, but also more interestingly, identify patients who completely meet their eligibility criteria, but did not respond to the treatment. So in this case, you can see, right, the red actually label these lung cancer tumors. And in this case, even you add a drug, even though the patient, express the tumor test the antigen, has the right haplotype, they do not respond to the drug treatment. And the tumors are still red, right? They're still alive. Now, we also allow them to test combinations. So one combo treatment. So one thing is that we found out a lot of tumors, they don't respond because they downregulated the HLA-2, which is the presenting complex that present these tumor antigens. So these tumors basically found a way to cheat. Now, by combining interferon gamma that boosts these expression, you can see that immediately, right, these tumors respond to the treatment and turning green, which is the cell death. So furthermore, right, to further be able to allow them to prove that, kind of this evasion mechanism. So another unique property of our mouse is that you can do direct infection of these patient mouse. Not like conventional bulk organoids, where you have to melt out the matrix gel, do single cell suspension, and all that stuff, which is very rough on the primary cells. In this case, we can do direct infection, and you can see this patient with the HLA-2, and immediately the tumor responds to the treatment. Again, showing that this is the mechanism that they need for the tumor cell to evade this therapy, and what we have to target the combo therapy to overcome. So basically, within 4 months, actually the major pharma actually now already filed FDA and are filing FDA and also IND for this treatment based on our data. And we also started, preparing for a clinical trial. Again, Duke is one of the sites with our assays, the companion assay, to select the right patient for this trial. So just, as kind of mentioned, right, I think this technology has a lot of promise. I think, there's many, when we first talk to the investors, they may talk about, organoid is a research tool, but it's just, we are dealing with live cells. It's never going to make it into a clinic. So, again, me, Dr. David, and also Hans Cleavers, we co-founded this company called Xylus, And this is actually, we got highlighted by the Nasdaq tower on the Times Square, which is very rare for a Series A company. But again, right, because we have, managing partner of Google Ventures, we have a UAE Sovereignty Fund, basically all have this one mission that, a functional assay has the potential to cover the 93% of patients that right now are not being helped by the genomic sequencing. So here, I want to, after the first part of the story, I want to kind of switch gear a little bit and also talk a little bit of more fundamental research in my lab doing to further understand better how the cancer cells transform in the body. So again, kind of a little background about colorectal cancer. So colon cancer is the leading cause of cancer death, as you all know. So the colon cancer advance in multiple stages. Actually, in early stage, you basically have adenoma. In those stages, actually, if they are identified and resected, the patient has great overall survival, 5-year survival. However, once the tumor cells start to become invasive in late stage, like 3 and 4, and also eventually become metastatic, right, the overall survival dropped significantly. For colorectal cancer, the 3 most popular sites is liver, metastatic site is liver, lung, and the lymph node. 70% of them first metastasize to liver, lung, you can look at about 20%. So at that time back then, my student Joyce, now she's a professor at the University of Chicago, did an interesting experiment with our collaborator Steve Lipkin and Weill Cornell, where I did a kind of the traditional subcutaneous xenograft model, but also did this orthotopic metastasis model, where formed tumor in the colon then metastasized to the liver and lung. And when we did this treatment, chemo treatment, what we found out is the xenograft tumor responded very well to the chemotherapy. The tumor in the colon responded a little less, but still oftentimes respond. But for certain cancer cell lines, interestingly, the metastatic tumor did not respond at all, or even have a worse effect in response to the treatment. And again, that's well known anecdotally in the clinic, where the simultaneously presented metastatic and primary tumor respond differently to the treatment. So actually, at that time, my postdoc Panchang, my student Quinn, did this experiment. They took the primary tumor and the metastatic tumor from our metastatic model and did a metabolomics. And interestingly, they found out there's a very distinct metabolic signature between the metastatic tumor and the primary tumor, even though they originated from the same primary tumor. And furthermore, if you look at a metabolic pathway that gets rewired, the several pathways really jump out. But one most curious is fructose metabolism, which before that I was never kind of paying attention to. So this is already published, so I just kind of keep it very quick. So what ended up with finding is when the tumor cells metastasize to the liver, and liver is actually a major site where fructose metabolism happen. So in the liver, actually, the liver environment upregulates this enzyme called aldolase B, which is a major fructose metabolism, and induce the fructose metabolism. And what that leading to is really shuffling the sugar into glycolysis and also pentose phosphate pathway. So what they do is, some of you have heard about Warburg effect, right? But what they do is, again, really boost the synthesis of biomolecules like lipids and also amino acids for cancer cell proliferation, while it doesn't actually shut off the metabolism that's going to the traditional TCA cycle, because the cancer cell doesn't burn as much ATP, right? So that become a way of fueling the proliferation of metastatic tumor cells in the liver. And actually, serendipitously, Pfizer actually saw our paper, and they just came up with the very first small molecule drug that target fructose metabolism, target this enzyme KHK, which is directly upstream of aldolase B. So we actually, I have been working with Pfizer now to bring this drug to potentially treat a colorectal metastatic patient. And furthermore, since our paper, there are a slew of paper again coming up showing the role of fructose in promoting tumor. So then we also asked a further question, which is that besides metabolic change, do these tumor cells also undergo more permanent change when they metastasize? So here we look at the epigenetics, more specifically chromatin accessibility, by performing this assay called ATAC-seq. How this assay works is you have transposons that label with a barcode, and the transposon will go in and insert them into open chromatin regions. So then by sequencing, you'll be able to tell which chromatin regions are open, so basically more actively transcribed, and what chromatin regions are closed. So here again, what we found out is, by and large, the metastatic tumors have the same epigenome as a primary tumor, but there's very distinct regions that were became open or closed if you compare the metastatic tumor versus a primary tumor. And there's regions that, while you're putting different, patient-derived cancers or cancer cell lines, very consistently when you go to liver, these regions alter. So you can see the blue is the metastatic, liver metastatic, and orange is the primary tumor. You can see they always alters when they go to metastasis. So this is the ATAC-seq peak, open chromatin peak. We further showed with a basic MIN chip with a ChIP-seq, showing that histone modification also changed in those peaks. And among those targets, we kind of became interested in a target called DPP4, and we checked with them with the human databases too, and confirmed that these genes really upregulate in the liver mets, also in human cancer. What makes DPP4 interesting is that it's not considered really a cancer gene. It's also known as CD26 and actually cleaves a lot of the peptides. But what's mostly known is it's actually a frontline diabetic treatment. So DPP4 inhibitor along with metformin is a two frontline diabetes treatment. So why does the tumor cells upregulate this diabetic gene DPP4, and what does it do to the tumor? So here we found out that if you knock out DPP4, you significantly reduce metastatic tumor growth in the liver and also prolong the animal survival. And furthermore, if we use CRISPR-Cas9 to target individual regions of these enhancers, all these four regions I showed you on that gene, you can see that in each case, there's going to have an effect on reducing the metastatic tumor. So these four genes, these four enhancer regions all contribute to the upregulation of DPP4 in the liver mets, and in turn promote their growth in the liver. So, and furthermore, if we use the frontline drug DPP4 inhibitor on these tumors, we can also see we have a similar effect of reducing liver mets growth. So, also very encouragingly, just a paper came out last year showing they did a huge epidemiological study where they found us that when diabetic patients with colorectal cancer taking DPP4 inhibitor, they exhibited a statistically significant survival advantage. And as we all know, right, like colorectal cancer, a cancer patient died from metastatic colorectal cancer, right? So all these patients, the survival advantage is almost all metastatic cancer patients. Again, consistent with our finding. And lastly, is most surprisingly, is the way DPP4 promote tumor growth is completely independent of their metabolic role for diabetes. What actually happens, DPP4 cleaves a peptide that regulates immune response to the tumor, more specifically neutrophils. So what happens is when a tumor cell metastasizes from the primary tumor to the liver, it actually being attacked by neutrophils in the liver because they came as a foreign cells, even though from the same body. But what DPP does is cleave the cytokine and reduce the neutrophil recruitment and attack of these metastatic tumors. So in this study, again, that's going to come out in advanced science, where the HGF hepatic growth factor in the liver actually upregulates a pioneer factor called PU.1, phosphorylate that, and PU.1 go in and open up those specific chromatin regions and then upregulate downstream factors like DPP4 and result in the growth of the liver tumor. And interestingly, both HGF, PU.1, and the DPP4, we actually have small molecule inhibitors to target them. So right now we are working to extend these preclinical studies potentially for treating metastatic colorectal cancer patients. But furthermore, another study we also embarked on using our epigenetic assay is also to study infectious disease. Again, I was being funded by this large DARPA program called Epigenetic Characterization and Observation, or ECHO. So the idea is, can we using epigenetic markers to develop better diagnostic and prognostic markers for infectious disease? So this study, we worked with Dr. Chris Woods, who is the chief of infectious disease here at Duke, where we went out and into the community and collect a large number of subjects and taking their serial blood samples. And then these, some of them actually contracted COVID-19, and we follow them along into the treatment. And in these specific cases, we are capturing very early time points. The day zero means early time points where the patient's seroconversion hasn't happened, right? So at that time, we don't even know if they got COVID-19. And then we capture on day 7 in the middle of the seroconversion when the blood started generating antibody against the COVID-19 virus, and at day 14 post-seroconversion. And then we later look at the patients who were healthy, who were exposed. Usually a spouse got COVID, patient showed a mild symptom, and patient showed a very severe symptom. And we did a both bulk and a single cell RNA-seq and ATAC-seq. So if you look at day 14, interestingly, if you look at a healthy, exposed, mild symptom, severe symptom, these patients exhibit very distinct epigenetic markers. And you can see this kind of progression from health to mild to more severe symptoms. Now, if we look at day 0, right, can we predict whether a patient will go on to develop mild symptom or severe symptom? So if you look at RNA-seq, just the transcriptome or gene expression, you can see that a lot of those, the genes are later on, but in early stage, you cannot really differentiate them between from the mild versus severe, right? So before seroconversion, before this antibody come up, we could not predict whether a patient would later on develop a severe or mild symptom based on transcriptomic gene expression signature. However, if you look at ATAC-seq, look at the chromatin accessibility, interestingly, there's very distinct markers actually differentiate those two cohorts. And you can see the cluster clearly tell them apart. Furthermore, if we do this kind of bivariate footprinting analysis, where we look at the chromatin regions where the transcriptional factors bind, you have to look at both the depth of the binding, but also accessibility of its flanking region. This is basically predict whether a region gets open by chromatin factors. And in this case, we actually identified that in mild versus severe, there are distinct factors that actually come in and open up chromatin regions in severe cases versus in mild cases. So if you look at those chromatin remodeling kind of events, most prominent, interestingly, in the early stage prior to seroconversion. Again, keep in mind, right now, we cannot tell them, right, these patients apart at all. But in that stage, what's really happening is CD14 monocytes. So these are monocytes that later can become macrophages, and the second, dendritic cells, right? The third is the CD16 monocyte. So these are all the innate immune system. So basically what we learned is in the early stage of COVID-19, it's really right that it's too fast. The adaptive immune system is still not able to respond. It's an innate immune system are responding to this COVID-19 infection. And depending on how strongly and fast the innate system can respond to the COVID-19, actually later on determines whether the patient will have a severe or mild symptom. You can see that in late stage, right, other cell types take over, the B cells, plasmablasts, rather than the monocytes. So when we did a single cell also analysis on those patients, we see distinct changes in the population, and we followed with single cell ATAC-seq. So long story short is we do this called a time trajectory or pseudo time analysis, and you can appreciate that. You can see that right from earlier on. So this is looking from mild and more severe. You can see clearly this progression of right in more severe cases, the different transcriptional factors that driving the chromatin remodeling. And if you look at chromatin accessibility, which is consistent with that. Well, if you look at gene expression, it happens kind of much later. And again, this is also looking at the time trajectory when a patient first convert from seroconversion negative to positive, again, showing how the chromatin landscape changed, driven by these transcriptional F pioneer factors that alter these immune cells, chromatin landscape in response to COVID-19. So again, here now we are on a, again, the ECHO program is further funding us to now developing a diagnostic, prognostic assay that for us to be using epigenetic markers to predict early on prior to seroconversion whether a patient has a risk to develop a severe symptom that later on require ICU and critical care. So now I'm going to switch to the third part, which is my lab has been studying on microbiota. So as many of you know, right, the microbiome research has been really hot in the recent years, and there's a lot of interesting studies. And this is kind of showing the progression of how microbiome research kind of caught on, really, kind of boosted by the NIH Common Fund. So there's many data, studies, data sets out there now on the microbiome distributions. However, one thing people don't realize is if you look at the majority of studies, right, these still on from the accessible side, like a feces, vagina, right, skin. However, we still know very little about what microbiome actually reside in the internal tissue or organ. You can see if things like, even the ileum, right, the small intestine or GI tract, the study is much fewer, right? Because we all know the bacteria in the feces does not necessarily correspond to the bacteria in the organ. So again, but how do we access right, what's happening in people's, in those internal organs from different patients? So there are large study data sets like a TCGA, the Cancer Genome Atlas, and a GTEx, where they did all this sequencing on human genome, right, from different organ sites. So my student Anders is thinking, can we leverage those data sets, but instead look at what microbiome are there? So here we leverage a pipeline that developed a while ago called PathSeq. What we essentially do is do the opposite. You throw in a human genome and look at what's left and try to map them onto microbial genome. So sometimes, I made a joke that, right, it's kind of basically we are rummaging the garbage can, looking at things people throw away to see who can find useful information. So that pipeline, when it's published, got a lot of attention because it recognized these bacteria called fusonuclear bacteria that have been shown to strongly correlate with colorectal cancer. And there's all these studies. But interestingly is, if you look at all the studies, fusobacteria is the only one that was ever discovered, right? So why we haven't discovered a lot more using this such powerful pipeline? The biggest problem is because the low biomass of this tissue and the contaminations. Because the microbiome field has been plagued by contamination. During the processing, as you guys can imagine, right, a lot of contamination can be brought on, so you don't know what's really came from the tissue or without the contaminants. Now, for feces study, it's fine because there's so much bacteria in the fecal matter that it dominates all the contamination. But when you go down to these low mass tissues, how can you tell them apart? Right, so studies saying, the publishing a method to examine contamination batch effect should be used. So here, Anders kind of came up with idea saying, look, can I use the sequencing from different organ sites, and by comparing them, right, and looking at the solid tissue versus the blood, comparing colorectal cancer, which should have a lot of bacteria, and brain cancer, which should have very little bacteria, and look at the sequencing data, whole genome or whole exome, and deduce and distinguish contamination from tissue-resident bacteria. And indeed, that's the case. You can see from colorectal cancer, we can get, or rectal normal or cancer, we can get a lot more bacteria. If you look at the brain or blood, they have a lot fewer. And furthermore, interesting, you can see, right, from the colorectal tissue, it's very distinctly different from the blood. While in brain, you cannot tell them the brain cancer from the blood, suggesting that they might all just be contaminations. And furthermore, what we took all these samples from TCGA and looking at and sequenced them, you can see there's the first principal component analysis clearly distinguish tissue sample from blood, suggesting that the bacteria from tissue and from blood is different. But we kind of was puzzling for a long time for the second component, right? What's separating those tissues on the second component? And what we found out is what's separating the second component is actually the sequencing center, where those tissues were sequenced. So what that's suggesting is that these are actually contamination brought on by the different sequencing centers because they actually get the same tissue source. So you can see that, right? If you look at a brain, you cannot tell brain tissue or cancer tissue from blood tissue apart, suggesting that, right, all these are probably, most are contaminations because there's no difference between those two. And furthermore, you can look at different bacteria, and there's a clear, sequence center signatures brought in by different sequence centers. So here, using this, using a statistical now, we developed a statistical method, but in principle, it's very simple, right? By comparing, for example, colon cancer with the blood, we can identify bacteria specifically in the tissue from the contaminations, which are equiprevalent, means they are in both blood and the tissue. In brain, as you can see, they are mostly just contaminations because they exist in both. And furthermore, it makes sense. When we look at our tissue resident, they have much narrow distribution on genome size, on optimal growth temperature around 37, right? While the equiprevalent has a much wider distribution, suggesting that they might came from the environment. And furthermore, the different sequencing center, you can see brought in, Broad, Harvard, and Baylor bring very distinct contamination signatures. And after our correction, we can isolate we can actually get rid of these sequence center specific contaminations, give us a lot of confidence. Now, with that, we actually went back to the original TCGA samples. So there's some source still kept the original TCGA sample, and we did independent validation, getting those samples. So as you can see, right, if we just get those samples and run this analysis, if you look at compared to 16S microbiome characterization, these different sequencing site bringing these contaminations. And if we do our correction, it corresponds much better with the 16S profile. And this also allow us to be able to now tell, for example, what are paired analysis to compare, right? So what are tissue-specific bacteria versus what are not tissue-specific. And furthermore, this finally allows to tap into these huge databases like TCGA, right? Again, finding out there's a co-abundance group of bacteria. For example, colorectal cancer patients that has fusobacteria tend to have this cluster of bacteria, while the patients have, the other patients tend to have more the bacteroides kind of clusters, right? Suggesting there's a microbial classification of these cancers. And furthermore, now the power of this approach now is that now allows to correlate the tissue, the microbiome, with also the patient's host tissue cell expression, including epigenome, genome, transcriptome, and the proteome. So here you can see we can perform this study and further show a strong correlation between the bacterial abundance with the host response, including RNA-seq and protein. And last is we developed this large database using the same method for multiple cancer types in the TCGA. For example, these 5, colorectal cancer, rectum, head and neck, stomach, and esophageal, you can see, right, these 10 cancers now surprisingly have a much stronger signature of tissue microbiome. And based on that, we developed online actually tools that has been now kind of heavily used by many groups to really try to dissect what are tissue-specific bacteria. So lastly, I'm gonna kind of speed up a little bit, just talk about the last piece of my lab's direction, which is the imaging. So again, as engineers, we are always very interested in what's happening in the tissue. But traditionally, we also always have a very static, right, picture. We take a biopsy, we do the histology, we do the screening. But we know these tissues are dynamic, right? There's dynamic homeostasis going on. So here, my lab has been kind of inventing several tools to imaging the gut in live. So my student Nikolai invented a scaffold coupled with abdominal imaging that allows to image the intestine live. And after that imaging, we collaborate with another faculty, Diego Borocats here at Duke, because he discovered that an interesting phenomenon where these neuroendocrine cells on the gut epithelium interestingly seem to form some kind of synaptic structures with the neurons. So he was wondering what those synapses do, which he terms a neuropod, right? By that time, the entire field saying, look, it doesn't do anything, right? It's all humoral, means these neuroendocrine cells just secrete factors. There's no physical link between these neuroendocrine cells to the gut, to the brain. So here, Diegos did a rabies tracing where he showed that when we put a rabies virus in that can only jump once, one jump, this virus already jumped from neuroendocrine cell to the nodose and to the nodose ganglion right next to the brainstem. So that suggests that there might be a very direct link from the gut epithelium that's sensing the nutrients and bacteria directly, and just one jump, it's already sending a signal through the nodose ganglion sensory neuron to the brain. But at that time, no one believed there's a function. So for that, we started collaborating by using both our window and also nice thing about intravital windows, we are allowing optogenetics. So the question is, if we stimulate these endocrine cells in the gut using the neurons, can we get a signal out from the nodose ganglion? So, and also my lab does some neuromodulation and stimulation recording too. So we also perform the vagal recording. So what we show here is if we stimulate them with the neurons like a glucose, we can get signals from the nodose ganglion all the way upstream. You can see here when we stimulate with sucrose. Now, interestingly is if we use optogenetics through the window, just stimulate this neuron with light, we get a very similar response from the nodose ganglion, showing that this is a physical direct link where the neuron gets activated by the nodose, the endocrine cell. And furthermore, if we use a halorhodopsin, which actually does the opposite to shut off this endocrine cell, you can see again, the sucrose can activate these neuroendocrine cells and we get a signal all the way up in the nodose. But here, if we shine light on the halorhodopsin, we shut off the neuroendocrine, we do not get that signal anymore, right? So this means that's both necessary and sufficient, these physical link from endocrine cells to the sensory neurons for the brain to sense these neural signals. So because of that, now I'm leading the Chen Zuckerberg single cell atlas, human cell atlas to map the gut-brain axis using single cell to understand this link. And furthermore, so that's great, but also in the gut, a lot of diseases happen in the colon, not in the intestine. You know, colon cancer, right? A lot of IBDs, Crohn's disease. So the question is that, can we also image the colon? That sounds easy, but the challenge is the colon moves a lot. So here, Nikolai invented a very, very smart device using ferromagnetic, where we have a scaffold that usually held loose. But if you block the colon, the animal's going to die. But during imaging, we use magnets to attract them so we can stabilize the colon. And we can image, things like, colon stem cell live for the first time. And also monitor immune response, for example, in this case, when we did add LPS, or we add a pathogenic bacteria, we can track them live. But one question we use a window to ask is, does sacral nerve stimulation truly talk to the colon? So what does it mean is sacral nerve stimulation, basically sacral nerve is a nerve that goes from spinal cord to the colon, and companies like Medtronic already have devices like InterStim that do the modulation, and that was FDA approved with many patients carry around. However, there's never proved that sacral nerve stimulation actually activates the neurons in the colon, right? So because for the neuromodulation, one challenge is you cannot tell apart placebo effects from real effects. When you stimulate, the patient knows, right? So this has a big controversy in the field. So here, again, using the colon window coupled with sacral stimulation, we showed for the very first time that we stimulate these neurons, we can actually activate, when we stimulate the sacral nerve, we actually activate the enteric neurons in the colon. So showing there's a mechanistic explanation of why this stimulation might work. And here just kind of quantification showing that when we stimulate, right, we can, and only a subset of enteric neurons in the colon actually responds. So this is already published, this slide, and respond to sacral nerve stimulation. And this allow the very first model allow us to actually screen by changing different parameters, sweeping the different modulation parameters to find the best treatment. And last is my postdoc Chang came up with a great idea. He said, look, it's in the abdomen, can we use it to understand embryos? So as many of you know, right, the embryologist and embryo development is a very dynamic process where from a little tiny embryo and that the organs develop. So now for very early stage from embryonic day 0 to 9, there's this imaging tools called the in total imaging where we can look at how what things happen. However, from embryonic 9.0, there's no way for us to image the embryo because now you require the placenta, and it's very difficult to recapitulate placenta-embryo interaction in the petri dish. But this is where the majority of the organogenesis, right, the organ formation happen. So my postdoc Chang kind of invented this embryonic window that for the very first time we can actually track the embryos during this development stage and look at how they develop. And these mouse end up actually growing fine. This one is the one that under image. You cannot tell them apart. And the mother is still fine. The mother carrying the window can still nurse the little baby mouse. So using this tool now, we can actually examine all these processes in the embryo. For example, here we look at it during the eye development. There's actually a lot of autophagy going on. And that's exactly correspond. If you take the embryo out and do the staining, you can see how similar the patterns are. This is for the very first time we image the neurotransmission in a nascent brain, where for the very first time, this neuron activated second neuron through the synaptic activated third neuron. So now provide a very first tool that we can study the formation of the neural circuits in the embryonic development brain. And furthermore, in this case, we also are able to characterize when we put a drug in, what's the kinetic for a small molecule dye to permeate through the umbilical cord into the embryos. And also we are able to, kind of showing if we do the viral, if you target the embryos using gene viral targeting, what's the dynamic of the virus that go into the embryos. In this case, we also, kind of image neuroprogenitor brain development, look at the neuroprogenitors that are dividing live in the live embryos. And last, we started working with Dr. Rudolf Janisch, who some of you know, right? He's the forefather who invented transgenic animals. So but Rudolfo and the whole field has been a long time trying to create chimeras, means putting interspecies embryonic cells into the embryos, and in the hope of the mouse will grow a kind of more humanized tissue, but for therapeutic development. But what they found out is oftentimes when you put a human from the human neurocrist cells in, these cells are nowhere to be found when you get embryos out. So what's happening to those cells? Because during the embryonic stage, there should not be any that they should be centrally immune tolerated. So to answer this long-standing question, we work with Rudolfo's lab, and here when we actually doing imaging, we show that in this case, when we injected a green labeled human neurocrist cells into the embryo, what we discovered is initially the red is a mouse neurocrist cells and green is human neurocrist cells. You can see initially they coexist, but the mouse neurocrist cells quickly expand and actually at the expense of the human neurocrist cells and basically eventually kind of eat them up, right? So this again showing this that and also if you look at individual cells, they don't have that dynamics. So what they show is in the mouse cells in the niche actually is going to outcompete and occupy the human neurocrist, human cell neurocrist niche. So actually my lab has now actually been developing a new very exciting technology that significantly enhance this chimera efficiency for human tissues. And last is we also invented the first window that we can image the placenta during the embryonic development. So for the very first time, understanding a lot of the diseases. So just to summarize, my lab kind of perform research in both functional omics, microbiota imaging, really to answer the central question about how can we understand tissue function better to improve precision medicine. So here, right, we performed the mouse technology to guide the precision oncology. We understood better metabolic and epigenetic reprogramming that provides new therapeutics and diagnostic opportunities. We provide a comprehensive decontamination algorithm to identify true tissue resident microbiota, and last, performing intravital imaging to capture tissue dynamics. With that, I want to thank my lab members, and again, thank you for the invitation to talk about my work. Thanks.
Excellent talk. Thank you so much. Let me see a few questions. Does the epigenetic marker vary with demographics?
So yeah, that's actually a great question. What actually we found out is that, so that's actually in the epigenetic field. So we have been doing a lot of studies on looking at the epigenetic drug resistance. So we grow organoids from different patients and target one drug and look at how the tumor become resistant. And what actually we found is from patient to patient, there's very significant differences, right? And they seem to be, they can go through different epigenetic methods, pathways. So in the metastatic case, interestingly, is all these patients all undergo this DPP4, but which is rare. In most cases, they actually undergo different methods, different pathways. So you're absolutely right that kind of compared to oncogene mutation drivers, the epigenetics seem to even present greater diversity that kind of providing a challenge for developing therapeutic drugs that targeting those mechanisms.
Thank you. Hans Clevers did a lot of this. One of the first to grow, this intestinal cells using stem cells. But then you started a company with liver. Are there plans to make, intestinal organoids and things?
Yeah, absolutely. Actually, I didn't talk today. So, mouse, we did several studies. We do a lot of intestinal organoids. I have another DARPA funded project right now, basically developing this using the intestinal mouse as a tool to identify pathogens, right? So in that project, because what happens is when they have, war fighters or soldiers going to the field, in the water, you don't know what pathogen you're experiencing, right? Like you can characterize them. So actually we're using this tool to really understanding the host intestinal and the pathogenic interaction. And also to your point, it's very interesting. When we do this mouse and we can actually flip the polarity. So a lot of you know, right? The epithelial cells have the apical side facing the lumen and the basical side, right? Facing the base memory and the blood vessels and the mesenchyme. And I didn't even think it mattered. But my student actually did this trick to flip. So we found out that in many cases, whether the bacteria come in contact with the apical side or basical side, actually have a very different response, right? So, but now from the hindsight, it makes sense. You can imagine from when we at LPS, right, the bacteria polysaccharide, when you put it into the apical side, it doesn't respond at all. When you put in basal side, the epithelium go crazy. But that makes sense because if it comes from the basal side, it means the bacteria got into the bloodstream, right? So you need a very strong response. While from apical side, you're going to have information all the time if you respond every time. But so we get very different. So we do a lot of intestine, we did normal liver. A lot of farmers are very interested in toxicity study, right? Using the mouse, high throughput mouse screening. But most exciting, I thought I had a slide in, but I probably took it out, was that we just recently we are actually submitting that manuscript too, side by side with our clinical trial and the immune study, is that we actually, from autopsy sample, we simultaneously generate this whole mouse atlas of the entire respiratory and the digestive tract from 5 different autopsy deceased patient, and also from 3 COVID patient who died from COVID, and basically using the mouse and study how the host respond to COVID infection. Going back to your question, the challenge about conventional bulk organoids is that it's too thick, right? The virus couldn't get in. You have to dissolve, become a single cell, and you have to disrupt the tissue structures. So in our cases, using mouse, we can actually do high throughput screening. So we actually screened for a lot of drugs. I found out some novel compound that seem to be very promising.
Thank you for the detailed answer. In your organoid model, are the cancer cells mixed with CIFs? If yes, is there a need to measure mechanical properties of the ECM to understand the activities of the CIFs?
Fantastic question. So in our model, yes, because if you look at, right, especially in our diagnostic model or immune model, because we do them within 14 days, usually, or 20 days of the samples, so all the cells are still there, and the calf is definitely there. And we can actually changing the stiffness of this ECM. We actually have a collaboration now. So some of you might know Roche has a new institute called the Institute of Bioengineering for Translation, or ITB. The head Matthias Luthoff, right, was at EPF, well-known biomaterials. So we're actually using our technology now, working with them, changing the stiffness using different cross-linker hydrogel to basically study how the stiffness affect. We actually already see very significant difference from when we look at the metastatic tumors. When we're changing the stiffness, the calf respond differently. The cytokine is different. And also, the tumor resistant to the treatment is different. So great question.
Excellent, excellent. One final question. Liver hepatocytes, they're not the same. Each liver hepatocyte is different if you do sequencing on the liver cells. When you're making organoids from different people, how do you deal with heterogeneity of these liver cells?
Great question. So first, liver, right? So if you look at a liver, traditionally we think all hepatocytes are the same, right? Hepatocytes can different, can proliferate, they can do all those things. So there are multiple heterogeneous. So I actually had a paper with NMADO and the JCI Insight last year. We did a very first single cell epigenetic profiling of liver during regeneration. Actually, even found out even within the hepatocytes, there's actually differences, right? During regeneration, there are actually cells that have more metabolic function. There are cells actually going back to more, progenitor state. So there's, and the second is the reason our mouse end up being very unique on liver toxicity compared to, as many of you know, right? Culturing liver, primary liver is very challenging, right? Even you look at Hunt's lab, had multiple, they have cell paper on the liver. If you look at what the lab right now mainly doing, they're actually doing fetal liver, right? They are not allowed to do adult, but it's very passaging. So there's several challenges, but one of them is when you do liver, cholangiocytes tend to take over, right? They're hepatocytes, but cholangiocytes are very, in the individual condition for some reason, it's very proliferative. So that has one major challenge. Using mouse, the great advantage is you are segregating these cholangiocytes, right, in individual droplet. So they're not taking over the entire population. It end up being a significant difference. But going to answer your question, and also in liver cells, as you said, there's also liver cells, we all know, right? Liver cells can be polyploidy, right? So liver cells can have multiple, chromosome, not just one, right? So if you look at this whole zoning region, right? So if you look at Shalav's scholars paper, right? They have there's 7 or different 9 kind of different, right, subclusters. So going back to your question, from liver to liver, there's definitely, first within the liver, there's all this heterogeneity we are characterizing. Second, and also the key things, don't let one dominant population take over, but also from patient to patient, great heterogeneity. And that's why the farmer right now, right, I think multiple farmers have initiative of replace the animal. Actually, quoting from some of the farmer, right, they said, look, when we do the animal, you always get a beautiful small error bar. It's all consistent. When you put in patient, that's like all over the place, right? So in our case too, like you only get a different liver. First is even growth is very different, right? Some grow great, some completely don't grow. And second is the drug treatment is often also significant variations. So that's exactly the reason that a human-based model potentially can complement the current animal-based model because we're just not capturing that diversity of human.
Excellent talk, sir. I appreciate your time. That's all the questions I have. Again, thank you for your time.
Thank you, Mohammed. Great, and thank you everyone for listening. I know I went over time and really appreciate it.
Great. Have a nice day.
Okay, thank you everyone. Take care.